fertility phase of mitosis, cytokinesis is what happens. Cyto means cell, and the kinesis part of the word is talking about splitting. So anytime you see cyto as part of a word, or cyte, it means cell. So cytokinesis is where we divide the cytoplasm and its contents. Mitosis is where we divided the nucleus in half. We took all the chromosomes and their copies, we pulled them apart, we made 2 nuclei. That's part of the cell, but everything else that's in the cell needs to be divided in half too. So when I say the cytoplasm and its contents, I mean the rest of the organelles that are in the cell, right? Cells have things like a Golgi apparatus and an endoplasmic reticulum and vacuoles. All that stuff needs to be divided too. We are not going to go into details about those structures, just that it's the other stuff that's in the cell aside from the nucleus. Now I put some pictures here just to illustrate things, but I just want to remind you, drawing pictures is not typically a requirement on any of my tests. You can always choose to if it helps you. Cytokinesis looks different in an animal cell versus a plant cell. And we're going to observe some plant cells and some animal cells in our little lab today. An animal cell during cytokinesis will look like what we've been looking at so far. Really distinct pinching of the mother cell to form 2 daughter cells. So if this is my elongated animal cell, the way that the pinching happens and then finishes is we have these little microfilaments that cause the cell to pinch and then separate. You do not need to be able to describe the mechanism through which this happens. You should recognize that it happens. In an animal cell, the mother cell pinches. That's how the cytoplasm gets divided. In a plant cell, however, it looks a little bit different. Plants, if you'll recall, Science 10, maybe even junior high, plants have a cell wall. They have a more rigid external structure, and pinching is not easy. Plant cells are also more rectangular than round. And so in a plant cell, instead of having that pinching, a little cell plate will form between the 2 daughter cells and eventually become a new piece of cell wall. It will not have pinching like an animal cell will. So, what's the big picture? Animal cell pinches, plant cell does not. The mechanism is beyond the scope of our course, right? So you don't have to be able to explain how the cell plate forms, just that that's a distinct difference between the 2 of them. Now, does anyone want to ask cytokinesis related questions?
Sorry, can I just
Sure. Now, we would have seen before, but after cytokinesis, the cell just goes back into interphase. So even though this is like the end of my description of the phases, the cell hasn't ended. It's just going to start the cell cycle over again, as long as a stimulus tells it that it should move on to the next phase. Okay. I can always come back to ones if you need them. Now, if we talk about the cell cycle, there are some real-world applications that I want you to be aware of. One of them has to do with stem cells. If you are looking for more information, because maybe you're like, hey, this is what my presentation could be about, stem cell research, there's lots of that going on. Page 527 of your textbook has a little bit more information and could be a jumping point for you. We will also talk about stem cells again at the end of the course when we talk about reproduction and babies and stuff like that. But for now, I'm just looking at their link to the cell cycle. Stem cells can replicate for a long time by dividing. stem cells are the ones typically that you have at the beginning of an organism's life, before they decide what kind of cell they're going to be. Stem cells can give rise to cells with specialized functions. Now, there are 3 different types of stem cells. One of them is called a totipotent cell. These ones can form all the cells in a human body, plus any extra embryonic. So extra means beyond. So any cells that go beyond or around the embryo. So if you know even just a little bit about babies, you probably know they grow inside of an amniotic sac, for example. That would be an example of an extra embryonic cell. The cells that go around the embryo and support it. So these totipotent cells that can form basically any cell that a human would be made of, we would only find them within the first couple cell divisions right after fertilization. They won't last long. So you think sperm fertilizes egg, it'll go through mitosis a couple times, and then after that, totipotent cells are gone, okay? Now that you also have pluripotent cells. Basically, all of the embryonic cells after those first few cell divisions would be called pluripotent. In an embryo, it's just a clump of cells for the first little while. It doesn't really look like anything. There's no distinct structures. It's just a clump of cells going through cell division. And those cells are all pluripotent cells. They can become any cell of the body. They cannot make the cells that support the embryo while it's still developing. So they can't make the extra embryonic cells, but everything else. Then, baby's born, maybe starts living a while, and then humans will still have what we call multipotent cells. You have some of these. You have cells in your body that could develop into potentially more than one type of cell, but they're limited. They can't be everything. So for example, you have bone marrow. In your bone marrow, blood cells are made, but you have more than one type of blood cell, and they can all be made in your bone marrow. They can't make liver cells, they can't make brain cells, but several blood-related things can all be made there, okay? So they're what we call adult stem cells. You don't have to be a legal adult to have them, but they're after embryonic development has finished, baby has been born. The other place that we often find multipotent cells is in cord blood. There is quite a big industry of places that will conserve cord blood when a baby is born in the event that that person, as they grow, requires some sort of stem cell treatment because they have a condition. That way they have some stem cells that are from their own body and would have the same DNA sequence and should be compatible. So they're called cord blood banks. That's a thing that's happening in the real world. Maybe that sounds interesting to you. So I'm just trying to give you some real world ways that these things could be applied. Now, does anyone want to ask anything about stem cells? Really, nobody has any comments here. This is a really good one for the ethics of when we should collect stem cells. Can we collect stem cells? Should we collect stem cells? Should we use them? What should we use them for? There is a lot of controversy about collecting and using stem cells. Perhaps you can see how, because embryos have stem cells, they are a good source of them, but that people might have very serious issues with taking embryos just for their stem cells. Like there's a lot of stuff going on, and so it can make a good topic for discussion. Now, Now the other thing that I want to mention is cancer. But I think here's what we'll do, because I can see some of you are trying to write and some of you have stuff printed. Last little piece for today, I kind of mentioned this yesterday already, but is cancer. If you're looking for a little bit more about cancer, page 560 of the textbook, which is in the chapter 16 document, if you're looking at the digital version, that's where you can find a little bit more about cancer. Like, it's not a ton, it's not exhaustive, but it can be a starting point if you think this is something that you want to research for your presentation. Cancer isn't really one thing, it's a group of diseases that are all associated with uncontrolled rapid cell division. So normally there needs to be a stimulus telling the cell when to do it, and in a lot of cases the cell is circumventing that or moving too quickly. Now in cancer, rather than spending most of their life in interphase, cancerous cells move very quickly from one cell division to the next. So instead of 10% of their life in mitosis, 90% in interphase, they'll have like mitosis, 10% of the time, mitosis again. So they're really going to shorten up the interphase part where the cell grows, builds structures, gets energy, all the stuff that it needs to do to be functional. And so without this extended period in interphase, the result is that we get this big, fast-growing mass of non-functional cells. And that's what we call a tumor. So those are